Hello everyone. This session is on mutations. It will be just a simple overview of what mutations are. Let us look at what the learning outcomes of this session will be. One will be able to comprehend what are mutations and what can be the causes of these mutations. What are point mutation and its types. One will be able to relate to something that is called as frame shift mutations. What are the implications of these mutations? One could explain using give a few examples. And of course, that mutations can be induced and has contributed to understanding the function of many genes. So let us look at what is mutation. Now, say for example, one is writing a sentence with each alphabet responsible for meaning of that sentence. So let's say, for example, we've written a sentence that says, I would like to talk with you. Now, say, for example, while writing that same sentence, instead of talk, what happens is instead of T, what is written is W. So what happens is, I would like to walk with you. So therefore, there is just a simple change in one alphabet, that is, the T is getting altered to W. Let's say, for example, you have another example where the sentence begins with DNA synthesis. While if to that synthesis two more alphabets are added, which means ES is added towards the end, then it becomes the DNA synthesizes. So the meaning of the two changes. For example, DNA synthesis could just be a noun, while DNA synthesizes, the word synthesizes becomes a verb. So the meaning is altering. So like we see in framing of the sentences where when even a change in a single alphabet can make a change in the entire meaning of the sentence, so can happen with DNA. We all know that DNA basically is a polynucleotide, two polynucleotide strands wound around each other, with every polynucleotide sequence having a sequence of nucleotides. And the sequence of the nucleotides is important because they store the information. So say for example, this over here is a DNA molecule that is double-stranded and both the strands are complementary. Now, it is known fact that in a cell, what is happening is something that is called as central dogma. So the information that is stored in the DNA is transcribed into an RNA, and that RNA can be translated into a protein. So therefore, for this DNA to be transcribed, it has to unwind. So the DNA can denature to form two strands, and using one of the templates, an RNA is formed. So this process of an RNA being formed from a template DNA strand is what is called as transcription. Now let's say for example, this RNA is released from the DNA. So you have it separate. This will again wind to each other. So the double-stranded DNA gets back to its original form. But you have now what is present as an RNA available. According to the genetic code, the codon is a triplet, which means that in this RNA, every triplet is going to code for an amino acid, which means UCG will code for one amino acid, GGG will code for another amino acid, and so on and so forth. So therefore, every triplet codon is giving rise to one amino acid. This is amino acid 1, 2, 3, 4, 5, and 6. So this is a 6 amino acid long polypeptide which has been obtained from an RNA, and that RNA has been obtained from the DNA. So you can see a direct relation between the coding in the DNA with the sequence in the polypeptide. Now, say for example, instead of T, the T changes to A, just like the T changed to W over here. Say the T changes to A. So when this strand is used as a template to synthesize the RNA, instead of you having the T, you having a over here, what will happen is now you will have U, because Complementary to A is present U, which means that now in this entire RNA sequence, one nucleotide has been altered. So the final codon has, instead of AAA, it has AUA. And in all possibilities, AUA may code for another amino acid, which means now the 6 amino acid long protein will have a 6 amino acid long protein, but with the sixth amino acid changing into another amino acid. That means that the meaning of this protein can change, because instead of the amino acid 6, now at this position you have the amino acid 7. So with this understanding, therefore, let us actually define mutation. So mutation is basically an alteration in the nucleotide sequence of a gene or of a DNA. That is what is termed as mutation. Now, it is, say for example, a very different scenario in which we are not sure whether the alteration leads to any great effect or not. Secondly, it is not necessary that every mutation is going to be observed in the protein level. Say, for example, the mutation, and especially in the eukaryotes, if the mutation is in a non-coding region, if it is in a non-coding region, you will not be able to see any difference in any of the proteins in that particular cell. However, if there is a mutation in the non-coding region and the non-coding region has a typical function, then maybe that function can change because of the mutation that happens in the DNA. So effectively, whenever we think of a mutation, we think of alteration of nucleotide, single nucleotide or several nucleotides in a DNA. So that is the understanding. Now, you can have mutations at the DNA level, just as we observed in the previous slide, or you can have a change even at the chromosomal level. Now, when it happens in the chromosomal level, it is called as chromosomal aberrations. So you get aberrations in that, and you have definitely a change in the sequence itself. So for example, you have this chromosome where this entire part is deleted. The chromosome is going to become shorter, which means that there is a definite change in the chromosome, and because of that change there is going to be a defined difference in the cell. Or say for example, you have a chromosome pair, okay, and you have translocation of one part of one chromosome to another chromosome. So say this is fourth chromosome, and a part of the fourth chromosome is translocated to chromosome number 20. Now originally the chromosome 20 was short, but with the translocation the chromosome 20 becomes longer, while the chromosome 4 has become shortened. So therefore there is an aberration that is happening in the, and this is what is called as a structural aberration. So this is what is happening at the chromosome level. But you can have this happening at just the DNA level itself, where you can have a deletion or an insertion, or it could also be a simple base substitution. Therefore, let us understand what could cause mutations. So mutations can happen while the DNA polymerase is carrying out replication. So instead of adding a correct nucleotide, if it adds a wrong nucleotide and fails to proofread or correct that mistake that it made and move ahead with replication, then the mistake remains in the DNA. If that is happening, then that is a change in the sequence. And so that is an error. So a mutation can arise due to the due to the replication or replication error. You may have physical agents. So for example, if you have UV radiation, a UV radiation can cause a damage in the DNA, can cause mutation in the DNA. Or say you have, for example, a carcinogen, so a mutagen that can change a particular nucleotide or particular nucleotides in the DNA, then again you can have mutation. Or say, for example, a virus is inserting its genome into the chromosomal DNA, then the insertion of the viral genome into the chromosome DNA is again going to bring about a change at the chromosome level. So if you have mobile genetic elements like, for example, transposition, then it can remove itself from one part of the chromosome and insert itself into another part of the chromosome. That too is a mutation. Now, interestingly, If the mutation is in the somatic cells, then it is called as somatic mutation. And these somatic mutations need not be going to the next generation. So it is not inheritable. However, if the mutation, because of environmental reasons or because of mutagens or anything, if the mutations are in the germ cells, so for example in the egg or in the sperm, then what can happen is you can have that mutation going to the daughter, to the progeny. So any mutation that happens in the germ cells are considered to be inheritable. But anything that happens in the somatic cells need not be inheritable. Secondly, suppose you have an embryo developing. So from a single cell, it goes to 2 cells and then to 3 cells, to 4 cells. Now, amongst the 4 cells, if one of the cells has a mutation, then any further cells that originates from that one cell will all have the mutation. But the other 3 cells which give rise to many more cells will not have the mutation. If that is the case, then it is called as mosaicism. Okay, mutation, which is mosaic, because you only have certain parts of the cells in a body having the mutation, but other parts in the body do not have the mutation. While if the mutation was in the germ cell, that is in the zygote itself, embryo itself, before it has divided, then all the cells in an organism will carry that mutation. Now let us look at types of mutations. Let us first begin with what is called as point mutations. So point mutations are when you have a change or an alteration only on a single base. So you may have base substitution, that is a base gets substituted with another base. Then that is what is called as point mutation. Or say you have a base pair added, a single base pair added, or if we have a single base pair deleted, then all of them are what is called as a point mutation, because it's only one point at which there is an alteration. Now, in point mutations, again, what you have is different types. So you have what is called a silent mutation. Now let us understand what do we mean by silent mutation. So say you, for example, have a DNA template. Using this DNA template, you have it getting transcribed to a RNA, and this RNA gets translated to its protein. So you have what is basically a polypeptide which is having serine, proline, tyrosine, lysine, arginine and phenylalanine. So this is the sequence of the amino acid in the polypeptide that has originated from this DNA. Now say for example, that same DNA, suppose there is a point mutation and the mutation is in the last A. So this A has now changed to G. So it has been substituted with a G. In such a scenario, the RNA that will be formed, instead of having U, will now have C. So you can see there is a change in a single base pair. Now, effectively, suppose you have all of these getting translated, but UUU and UUC, they code for the same amino acid. Why does this happen? Because it is observed that one amino acid can be coded for by more than one codon. And fortunately, in this case, a UUU codes for a phenylalanine, but a UUC also codes for a phenylalanine, which means that effectively you do not have any change in the amino acid sequence in the protein, although there is a change at the DNA level. So when you don't see a change at the protein level at all, because of a point mutation in the DNA or in the gene, then such mutations are what is called as silent mutations. Okay, one more fact before we go beyond this is that suppose you have an alteration such that, or a base substitution such that a purine is getting substituted by another purine, then that is called as transition. But say for example, a purine is getting substituted by a pyrimidine, okay, then that kind of alteration is what is called as transversion. So point mutations can either be transition or they can be transversions. Okay, here what you have observed, A getting substituted by G is an example of transition, but you can also have A getting substituted by C. So it remains CG, and you may so happen that UUG also codes for phenylalanine. In that case, again, that would be a silent mutation. So effectively, what does one understand by silent mutation? That there may be a change at a single base pair in the DNA, but that change is not reflected in the protein. And if that is happening, then that is what is called as a silent mutation. Now we go to the next point mutation type, or what leads to a mutation is what is called as missense mutation. So one of the types of point mutations is missense mutation. Let's again say, for example, you have the DNA template on which the RNA is being synthesized, and that RNA being translated to a protein. So the sequence of the amino acids in the polypeptide is this. Now, say for example, you have one nucleotide changing, okay? So you have this A substituted with G, but at this position. Then what can happen is that the RNA that is going to form will now no longer be UUU, but it will be CUU, okay? Now, so therefore this is one triplet, second triplet, third triplet, fourth, fifth, and sixth. But this CUU is no longer coding for phenylalanine, instead it codes for leucine. So if it codes for leucine, what is going to happen? The sequence of amino acids in the protein is going to be altered. Now, because of that change in one amino acid, you may have the entire meaning of the protein or entire function of the protein, or at least part of the function of the protein getting altered. So, therefore, when you have a change at the protein level in such a way that its functionality is affected, then you have what is called as a missense mutation. So the best example for this is the sickle cell anemia. All of us know that when the sixth amino acid, which in the original globin is glutamate, and this glutamate is actually substituted for by valine because of a point mutation. So the presence of valine changes the conformation of globin. And because there is a change in the conformation of globin, you see a change in the structure of the RBC at all. So you will see that the RBC becomes sickle celled. That's why it's called as sickle cell. But the second thing that happens is, because the globin has a valine now, it is also affecting the ability or the potential of oxygen binding to the globin. And that is why people having sickle cell or people having this defective globin will have an inability to bind to oxygen effectively, and therefore such people will suffer from what is called as anemia. So therefore, the missense mutation that you talked about is a perfect example of what you see in sickle cell anemia. A change in just one amino acid. The change in this one amino acid is just because of one point mutation, and therefore you have an entire organism getting affected because of a globin being different. And that too just because of a single mutation. Now let us go to another type of point mutation, which is called as a nonsense mutation. Now let us understand what is nonsense mutation. So again, take for example, you have the DNA template. Using that, you have the RNA being synthesized, and using that, you have the protein being synthesized. So therefore, this is basically the sequence of amino acids in the polypeptide. Now say for example, you have an alteration of a single base pair. So this A over here is getting substituted by C. Therefore, in the RNA that gets transcribed, you will have a G opposite the C, so which is complementary. So instead of a U over here, you have a G over here. Now, therefore, when translation happens, it is going to read AGC as one codon, CCC as another codon, and the UAG as another codon. However, interestingly, UAG is a stop codon. Therefore, the ribosome will not be able to add any amino acid over here because UAG does not code for any amino acid, which means that at this point, translation will stop. And because translation stops at this point, none of these will be present in the protein. So the protein will only be a dipeptide standing at serine and proline. That means what is definitely happening is that there is a change at the protein level to an extent where the protein is truncated. It has been shortened. Why? Because of the stop codon being present right in the middle of the gene. And this codon being present, stop codon being present right in the middle of the gene is because not maybe not even middle, maybe just 1/4 in the first 1/4 part of the gene. So you have a very small peptide being formed. So such codons or such changes which lead to a stop codon being present, those kind of mutations are what are called as nonsense mutations. Now let's look at another type of mutation which is called as frame shift mutations. As we go ahead, you will understand what we mean by frame shift. But frame shift mutations can happen because of deletions. Deletion can be of a single base pair or several base pairs or even long DNA. Okay, so say for example, let's consider you have again a DNA template. Using that, you have the RNA being synthesized, and using that, you have the amino acids present in a polypeptide. This is the sequence of the amino acids that are present in the polypeptide. Say for example, what has happened is that where you had an ATA, okay, so you had an ATA, so you had TATA, TATA, correct? So you have the same thing here right now. But let's say for example, you have this entire base pair deleted. If the entire base pair is deleted, right? So after transcription, your RNA will be one nucleotide smaller. Okay. Now, if this is one nucleotide smaller, what is going to happen is this is the first codon, which is the same over here. This is the second codon, which is again same over here. This is the third codon. Here you will find a change where you had AUA because of the presence of the TA. Now the TA not being there, what is going to be read is AU and this U now. So the AUU. So the next codon is going to be AUU. Okay, and thereafter the entire reading frame changes because what was UUU earlier now will be read as UUG. It has shifted by one nucleotide. So you will have, instead of GCC, which is read over here, you will have CCA. And then if it continues, you will have AAA, correct? So therefore, or AA, whatever, X, okay? So therefore, what you observe is from the point where deletion has occurred, the reading frame has changed. Reading frame of what? Reading of the codons is going to change. See, because in genetic code, what is observed is that the genetic code does not have a comma. Since it does not have a comma, it is going to be continuously read as triplets, 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 triplets. So whatever is the next triplet is going to be read. And that's why there is a shift in the reading of the frame. That's the reason why it is called as frame shift. So if you were to look at that, you would have this as serine, this as proline, but now thankfully AUA and AUU are still in isoleucine. So you still have ILE present. But look at the fact that what was UUU is now UUG, and UUU and UUG code for a different amino acid. So you will now have a leucine present over here. You will then have a proline, then you will have the next amino acid. So from this point on, you have the entire sequence of amino acids altered. So you can see that what began correctly as SPI, because of the deletion, now the reading frame has changed, and so the amino acid sequence has changed. So you will have a change in the reading frame, and because of that, you have a change at the protein level. So what has affected the DNA will be affecting the protein as well. Interestingly, if you have the deletions or insertions, deletions in multiples of 3, then it is not going to lead to a frame shift mutation, although it is still going to be a mutation. So we will look at that again. Now let's consider its insertion. So instead of a base pair getting deleted what can happen is a base pair can be added, or a few base pairs can be added. So here, this was the DNA template. You have the RNA template, RNA being formed, and then you have the amino acid sequence. So that is this. Now, say for example, at this point, you have a TA getting inserted. So what was TA now stands to be TATTA. So a T has been added extra. So this is what is called as insertion. So a base pair has been inserted. Now, it's not necessary that you only have a single base pair being added. You can have two base pairs being added. It is still insertion. So when you have extra nucleotide base pairs being added, then it is what is called as insertion to an already existing DNA. Okay. So therefore, what is going to happen is, over here again, you will have a change in the reading frame. This will be the first triplet. This will be the second triplet. This is going to be the third triplet. And now because of this, the fourth triplet is going to be AUU instead of UUU. So let's see what happens. So you have serine, proline, and isoleucine not altered, but from this point on, again, what you have is a change in the sequence of amino acids. So therefore, you can see that there is an alteration in the entire amino acid sequence after this point. So that point is the point where the insertion has happened. So therefore, the reading frame has changed. A change at the protein level is evidently observed. So we look at, suppose you have an insertion of a triplet. So say for example, you had AGC, CCC, and so on. Now you have AGC, CCC, and let's add another triplet, CCC. So you have what is called as duplication of this entire thing happening, but it is a triplet. Okay, or you may have this entire 6 getting added again, so then it is a 6-mer, that is a multiples of 3 being added. If that happens, the reading frame is not going to get altered. Okay, so this will still be AGC, UCG, GGG, but now you have an extra GGG, and then you have again AUA. So UCG, GGG, AUA, right? The reading frame from here is AUA. The reading frame from here will still be AUA. Okay, so therefore, what happens is that you only have an additional amino acid being added. So it is a mutation. There is a change in the amino acid sequence. There is a change at the DNA level as well. But since it is a triplet that has been added, the reading frame does not change. But during translation, this 3 is going to be seen as one amino acid, and that extra amino acid is being added. So a frame shift will not happen when you have triplets or multiples of, when you have a triplet being added or multiples of 3 being added, then you will not see a frame shift, but it is definitely still going to be a mutation. That can be considered as insertion as a whole, but it need not necessarily be frame shift. So although this has been put under frame shift mutation, you don't really see a frame shift over here. Now we look at something that is called as inversion. So this is the same thing that we saw earlier, where you have the template, you have the RNA is being synthesized into the amino acid sequence, forming a polypeptide. Now say for example, CCCTAT is inversed. So it becomes TATCCC. So the RNA that is going to form is going to have a different sequence compared to this. And so when you have the amino acids being added during the translation process, what you will observe is that you have a sequence that is getting altered. Because this was inverted, you have the ILE present over here and the proline present over here, as you can see. So due to insertion, again, the sequence of amino acid in the polypeptide is changing. So there is a change at the protein level happening because of what is called as inversion. So let us just look at, what are the general effects of mutations. Well, before we go into the effects per se, please understand that mutations can be random. That means the mutation can happen anywhere in the DNA. So that is called as random mutation. Or it could be specifically at a particular part or point of the DNA. If that is happening, then it is what is called as site-directed mutation. It can be induced. That means you can expose the DNA to a condition where the DNA will undergo mutation, or it could be just simply natural. Okay, so these are certain things that are associated with mutations. Another thing that is associated is that mutation can be reversed or nullified. That is again another thing that can happen. Now, what can be the effect of mutation? Well, a mutation can lead to gain of function. What does that mean? It means that a mutation will only help in gaining a certain functionality. If that is the case, that gain of function can be a positive sign for the cell or for the bacteria, or say for even the eukaryotic organism. Or it can be a loss of function. If it is a loss of function, again it may be good or it may be bad. We do not know. It depends on which gene, and it depends on what kind of gain of function and what kind of loss of function. So therefore, this is that a mutation can either lead to gaining a functionality or losing a functionality. Say for example, consider that E. coli can form infection. Okay, so it has pathogenesis. Now say for example, the, the E. coli is present in an environment where it needs to adapt. To enhance its pathogenicity, suppose there is a mutation, and because of that mutation, the E. coli is able to increase its pathogenicity, then that mutation will be consolidated because the E. coli has benefited from that mutation. So then that is what is called as gain of function. Now, if you have several such mutations happening over a period of time, then you may have a very new bacteria being formed from that original E. coli. So therefore, you can have over a period of time accumulation of several mutations leading to evolutionary changes. This is something that has been observed. So mutation is considered to have a role to play in adaptation, considered to have a role to play in evolutionary changes. It may have a great effect or it may have no effect at all. So as one can see, if it's a silent mutation, there is no effect at all. But if, say, for example, you have a base pair where you have the globin changing its conformation to lead to what is called as sickle cell anemia, then there is a big effect. So it can have great effect or it can have no effect at all. So these are some of the effects that mutations can have on the organism at the, okay, again, it can be just at the DNA level, it can be at the gene level, it can be at the organ level, a cell level, tissue level, or it can be even at the organism level. So you are looking at the affectation of the mutation at various levels. It can be just at the DNA level, or it can be at the gene level, or it can be at the organism level as well. Okay, so that is something that makes mutation a very interesting study. So let us just conclude. An alteration in the nucleotide sequence leads to mutation, and also if they are huge portions of the chromosome changing or even substitution of a portion, then they lead to what is called as chromosomal aberrations. Single base substitutions are known as point mutations, which can either be silent, missense, or nonsense mutation. Frameshift mutations are due to indels. Indels is basically insertions and deletions put together, due to which there can be change in the reading frames. Inversions too can change the sequence of amino acids in a protein. The cause of mutations can be various. It could be due to errors in the replication. It could be because of physical and chemical mutagens. It could be because of viral infections. Mutations, as mentioned, may have a great effect or not. And plus, not all mutations are inherited. So mutations can be random, can be induced, can be at the DNA level, or its effect can be at the tissue level, organ level, organism level. It can be somatic or it can be inheritable. Thank you.